Computer modeling is a tool, not the tool. The model is only as good as the assumptions put into the model. It has been clear from the start that the modelers have no idea of how a virus works in the natural world. They have based their modeling on the assumption that the culprit is a human being. The human being must be controlled in order to control the virus. This is completely wrong. Bacteria and fungi, at the cellular level, exist at the micron scale. But, they have the cellular machinery to grow on their own, i.e., their cells will divide and multiply as long as they have nutrients. We cannot see them normally without a microscope. But, if they keep growing, eventually we can see them, as things such as moldy bread, or mildew on the wall, or even feel them. Old vegetables that get a slimy feeling actually have a bacterial plaque on their surface. Both bacteria and fungi can form spores to protect themselves under harsh conditions. It is a form of hibernation. We have bacteria and fungi in our bodies constantly. Our immune system usually keeps them at bay, or more accurately, keeps them in balance. However, if our immune system weakens, or if a balance is shifted towards the bacteria fungi, the balance can tip in their favor and we can experience disease. We tend to have more difficulty with control of bacterial fungal infections than viral infections. In fact, the most common cause of a fatal outcome due to viral infection, including coronavirus, is a bacterial infection. The reason the second week of infection is considered the worry stage is not because of the virus, rather this is the time when a weakened immune system, either by exposure or by losing the balance battle, cannot prevent the bacteria fungi from taking off. Most people who die from influenza, coronavirus, even rhinovirus, do so primarily from pneumonia, bacterial infection, or some other systemic bacterial infection. Other things, besides fighting a virus, can weaken the immune system. Aging, diabetes, obesity, liver disease, kidney disease, cancer, lung disease, other infections, viral/bacterial/fungal, stress, circulatory problems, cardiovascular disease, and several others all can cause weakened immune systems. That is why they are called comorbidities. Clearly, the number and degree of conditions that weaken your immune system greatly increase the risk of severe disease or death from any infectious disease, bacterial, fungal, or viral. All of these things occur at a level where our senses cannot perceive them. Fortunately, our bodies recognize these things at the molecular level, and it is our own chemistry, we call biochemistry, that intervenes, mainly in the form of our immune system. The virus, what are we dealing with? My doctoral degree is in organic chemistry, specifically, chemistry involving carbon-based compounds. Chemistry is about working with problems at a molecular level. Guess what a virus like coronavirus is? It is a complex organic molecule. Organic chemists would call it a macromolecule, where macro means large. It is only considered large in comparison to small molecules. I am naturally inclined to look at a virus like coronavirus as an organic molecule. Coronavirus, CV, and influenza, IF, are very similar at the molecular level. Both are ribonucleic acid, RNA, viruses, and both are enveloped helical, meaning that they have a similar three-dimensional structure with a protein outer part and the RNA inside. CV is a positive strand RNA and IF is a negative strand RNA. This means they have opposite structures, much like you have a left hand and a right hand. Their viral class identification is different partly for that reason. Both CV and IF behave almost the same outside of the body, and this is due to their size, structure, and relative chemical similarities. Because of the small size, we really do not know how they truly exist in the environment. They could be floating around as individual molecules, i.e. as single CV/IF particles. They could aggregate, meaning that they form clumps of molecules, again, too small to detect. They could attach to any other particle in the environment. Since they are so small, they could hitch rides with dust particles, pollens, leaves, just about anything that they may have an affinity for. The list of possibilities extends to anything you could think of in the environment, including living creatures. In short, they simply could be anywhere and everywhere. In an outdoor environment, we know that the CV/IF molecule will start to break apart within minutes or maybe last an hour or two. The local environmental conditions will determine how fast the molecule breaks up. We know that heat and ultraviolet, UV, radiation are pretty good at breaking it up. There are things that chemically will help break it up. For example, saline conditions, like in an ocean, are good. It may be considered a natural disinfectant. There are man-made disinfectants such as bleach. We know that CV/IF are not stable under pH of 3 or over a pH of 10. So if the molecule encounters either natural or man-made conditions that deal with these pHs, the molecule will break up. Common soaps are good for breaking up the molecule. This is why there is the recommendation to wash with soap and water. Likewise, there are conditions that increase the stability of the molecule. Both CV/IF survive longer under colder conditions. This is probably one reason why they tend to favor winter months and colder climates. We know that certain types of surfaces can make it more stable. For example, CV has good stability on plastic, half-life of almost 8 hours, and has even been detected up to 1 week on surgical masks. Some types of metals, such as copper, can speed up decomposition, and some metals lend stability, such as stainless steel. Skin can actually be good at destabilizing because of not only sweat but also the natural oils and detergents that are produced in the skin can break apart these types of molecules. That is a reason that skin absorption is not considered a vector of infection. Serious breaks in the skin, however, such as from burns or injuries, could lead to infection due to the decreased natural inhibition. So, in general, we would want to try and increase exposure of the molecule to conditions that destabilize while trying to minimize the stabilizing conditions. The virus in disease transmission. The rationale for lockdowns, masks, distancing, etc. all rest on the assumption that human direct transmission is the greatest risk for disease. Anyone, at any given time, in any place can pass the virus to another. It sort of reminds me of the character Kofi in the movie The Green Mile. People seem to be convinced that somehow, the only way to catch this virus is because it makes a beeline from person to person. In other words, we are the culprits, but, is this really the case? In short, no. And here is why. Because of the modeler's view, if we imprison people, lockdown, a term used in penal institutions when prisoners become unruly, cover their faces, masking, and keep them from doing what people do, i.e. socializing, distancing, we can stop the virus. This concept is what wannabe dictators all over the world have embraced. This is nonsense. Certainly, you can get infected that way, but that is only one way of many ways. It may not even be the main way. It is losing sight of the forest for the trees. To examine the path to infection more closely, let's make the following assumptions, which you can see are more or less worst-case assumptions. Assumption 1. A person has CV/IF and is shedding, i.e. releasing virus from their bodies. Further, let's focus on the nasal/oral route for shedding as the only route, even though we know that the virus can be shed from feces. Assumption 2. All shed virus is infectious. This may sound like a strange assumption, but we really do not know how infectious shedding viruses truly are. What is being shed could be combinations of fragmented virus and more intact virus. The reason it is not clear is because the main method that is used for identification of samples is PCR. PCR cannot tell whether what is being amplified is actually infectious or not. When we exhale breath, speak, sing, laugh, cough, shout, sneeze, hiss, scoff, grunt, etc., air is expelled from our mostly upper respiratory tract. This air may or may not contain particles of moisture, mostly water. These moisture particles may or may not contain mucus, cellular debris, bacteria, etc. from our respiratory tract. These moisture particles may or may not contain virus particles. In other words, there may be virus particles hitching a ride or there may be none. There is no scientific evidence that when a person is infected that they are continually expelling virus, but that goes to a different essay. Please note, I am not referring to the playground use of the spitball, which is a massive collection of saliva, which may or may not contain any of the above. However, I think that we all can agree that amorous kissing when there is an infected person involved runs the highest risk of transmission. But this has more to do with direct contact. I want to deal with indirect routes of transmission. Aerosols and droplets, after leaving the mouth/nose, will quickly lose their moisture, i.e., the water base will evaporate. The smaller the particle, the quicker this will happen. With aerosols, it may be within a fraction of a second. Environmental conditions will also affect the timing. Warmer and drier conditions will speed up evaporation, while colder and more humid conditions will slow it down. Studies have indicated that under most normal temperature conditions, aerosols and droplets less than 100 micron in size evaporate before they hit the ground. What happens to the hitchhiking virus? It is still there. It does not evaporate. It has lost its ride, but it is still there. What happens to it now? It can go anywhere, i.e., it can be dispersed just like the free molecule. It will last as long as it is stable. It can be carried by the wind, outdoors, or by air movements or HVAC, indoors. It can hitch a ride with other carrier things, outdoor examples such as above. It can land on surfaces, any surface, whether indoors or outdoors. Animals or even insects can carry the molecule if it lands on them. If it lands on another person, it can land on their clothes, hair, skin, etc. and be carried by them. If it happens to get sucked into the respiratory tract or absorbed on the eye, it may eventually lead to infection if it can survive the body defenses. The possibilities really are endless. Indoors, the picture becomes even more complicated because now the vectors of movement, displacement, and contamination possibilities increase. Air handling units can redistribute the molecules to other areas far from the original source. Surface contamination is now a real consideration. Simple items can become sources of infection, for example desk pens and pencils, office equipment, telephones, notebooks, furniture, electronic devices, cups/glasses, dishes, light switches, etc. Just look around the room that you are sitting in and remember about when you, or someone, dusts. At least anywhere that a dust can go, so can a molecule like a virus. In fact, the very act of dusting could reintroduce the molecule back into the environment. Anything in that environment that you touch is a potential source. It should be easy to see why a lockdown is disastrous. A single sick person can spread a virus throughout a whole building and no one would know it until too late. Clearly, air handling, sanitation, people movement, shared items, all will play a significant role in transmission risk. Further, indoor conditions are better generally for stability and survival of the molecule. Why are meat processing/packing plants at risk? They are refrigerated facilities. There are many people, so there is a lot of movement. There are many surfaces for the molecule to sit, like carcasses, that are handled often and routinely. I think people can start to see the problem that we are dealing with and why the virus doesn't just go away so easily. The idea of masks on people did not suddenly appear in March of 2020. The usage of face protection with infectious diseases has been well studied, especially with influenza. Do not forget, the mechanics of these two viruses, CV/IF, are essentially the same, so what works or doesn't work for one is the same for the other. The understanding has been that a mask, and that term usually refers to either a surgical mask or N95 mask, has no benefit in the general population and is only useful in controlled clinical settings. Further, it has been considered a greater transmission risk than a benefit in the general population. If people still have a memory, you may recall that this was still the advice in February 2020. That understanding has not changed, and I will explain why. The term mask by itself means nothing. It is like saying car. You have to identify it more specifically because there are many different types and varieties, just like cars. So, for this essay, I will use two terms as follows. Face coverings. In this category I will include homemade cloth, dust, non-fitted utility, custom stylish, and any other common mask, i.e. something that is intended to cover your mouth and nose and that is by and large used in the general population, because they are cheap and inexpensive. Mask. In this category, I am referring specifically to the surgical mask and N95 mask, which is recommended for use in clinical settings by healthcare workers. If necessary, I will specify between them. One of the big mistakes by modelers is the concept of a face covering or mask as a barrier. I see many references to so-called experts who make this claim. This is completely false. No face covering or mask is a barrier. Either they do not know what they are talking about or they are misleading people. Masks and face coverings are filters, not barriers. They filter only the things that they are designed to filter, to a level of efficiency based upon design, usually not at 100% efficiency. For example, the N95 mask is designed and rated to filter particles greater than 300 nanometers at 95% efficiency. Note, there are masks with greater efficiency than 95%, such as the N99, but these are very expensive. I understand the psychological crutch that people feel with something covering their mouth and nose. I'm sorry. But that is a false sense of security. Perception is not reality. The mind says that you have some solid thing covering your mouth and nose, but that is not really the case. It is porous, things get through or go around it. I could spend time on the viral transmission and effectiveness of the variety of face coverings and fitted masks based upon the material, pore size, non-fit, etc., as well as the studies. I will say that there has been only one type of mask, the surgical mask, which has shown any ability to reduce, not eliminate, virus transmission because it is actually rated to a 100 nanometer pore size. But the surgical mask is not intended for use outside of a controlled sterile hospital surgical field, where its use and function can be controlled. It has limitations. So, what happens when a person wears a mask or face covering? There are two different views of how the mask operates depending on whether it is ingress, protecting the wearer, or egress, protecting the environment. But both add up to more or less the same thing. First, what happens on egress? We will look at droplets because most face coverings will not stop an aerosol and the 2020 propaganda has been focused on droplets. Assuming that a person is shedding virus and they produce droplets that contain hitchhiking virus, and assuming the face covering actually stops all droplets, best case scenario, the following molecular pathway will likely occur. 1. The droplet will lose its moisture. The timing may be different than just going out into the environment, but moisture will be lost. However, the expelled droplets may accumulate faster than evaporation. When that happens, the facial covering starts to become saturated with moisture, mucus, cellular debris, bacteria, as well as virus molecules. 2. The virus molecule does not evaporate, and no matter what happens as far as the droplet is concerned, the virus is now on the face covering, at least initially. This means that the face covering is now contaminated and is a possible source of transmission, both contact and airborne. 3. The virus is not somehow magically glued to the mask but can be expelled, whether or not there is still moisture. This can happen the next time a person breathes, speaks, coughs, sneezes, hisses or grunts. So, the virus can be expelled out into the environment from the face covering. So, the face covering acts as an intermediary in transmission. It can alter the timing of the virus getting into the environment, but it now acts as a contact source and airborne source. Virus can still get into the environment. Since we know that the stability is good on most covering and mask materials, it does nothing to break down the virus until the covering is removed and either washed or discarded, appropriately. Here is an important point: as more virus molecules accumulate, more are expelled. The face covering is not some virus black hole that sucks the virus into oblivion. Second, what about ingress? What works for egress works for ingress. So, if a person is wearing a face covering and they encounter virus, aerosols, or droplets, the virus and aerosols will likely penetrate. If the droplet is stopped, the surface is now contaminated. This means that if the surface of the covering touches the mouth or nose, you can become contaminated, i.e. infected. This is a common sight with most face coverings, including the stylish coverings that people are wearing. I often see the covering moving back and forth against their mouth and nose even as they breathe, like a diaphragm, as well as with the cheaper dust masks and homemade cloth masks. If you inhale, you can become contaminated. If you touch the face covering, such as pulling it up and down, you can become contaminated. Further, because the surface is contaminated, a person can also expel the virus back out into the environment just as with egress. This can be done by talking, breathing, coughing, etc. Stopping a droplet is not the same as stopping the virus. This molecular evaluation only assumed the best case contact scenario, that is, 100% contact between the face covering and any virus particle that may be encountered. I have not examined low efficiency coverings, inappropriate use and handling, non-fit. Air will circumvent the covering and go around it since airflow follows the path of least resistance, where the air goes so does a virus. I have not examined the eyes or ears as entry points. I have not examined the other modes of molecular movement on the surface of face coverings, such as osmosis. I have not examined the almost 100% misuse of any covering by the population at large simply because they have not been trained and have been misinformed and are using ineffective coverings. It boggles my mind when there is some notion that by wearing a face covering you are actually doing a service to your neighbor and therefore everyone has to protect everyone by this. Actually, the opposite is true. You are now becoming an additional potential source of environmental contamination. You are now becoming a transmission risk. Not only are you increasing your own risk but you are also increasing the risk to others. This is an excerpt from an article written by Roger W. Coops, The Year of Disguises, published October 16, 2020 by the American Institute for Economic Research. Thank you for watching this video. Please feel free to share this with your friends and family members.